friends, you are welcome in video lecture series on Bryophytes. So today I am going to discuss the next type from Bryophytes. Name of this type is Funaria. So I am going to discuss this type with reference to these points: occurrence and distribution of Funaria. Next one, external morphological characters or external morphology of this Funaria. Then anatomical details about the main axis of the Funaria plant, anatomy of leaf, and finally I am going to discuss the methods of reproduction in this Funaria. So if we see the occurrence and distribution of this Funaria comprises about 170 species throughout the world. Out of these 170 species, near about 15 species are recorded in our country, in India. Out of these 170 species of Funaria hygrometrica is a cosmopolitan species of Funaria. It is worldwide in distribution. It is found in all countries in the world. That's why it is known as cosmopolitan species, Funaria hygrometrica. Then, if we observe the occurrence of this Funaria, the Funaria generally occurs in rainy season, during rainy season, on the wet soils as well as on the wet rock surfaces, as well as in the crevices of the rocks, as well as on the poles, on the bricks, on the walls during rainy season. So this Funaria very commonly occurring during rainy season. So if we observe the morphological characters of this Funaria, the main plant body of this Funaria is a gametophyte, and this gametophyte shows two stages. The prostate stage of gametophyte is commonly known as protonemal stage, and erect stage is commonly known as leafy stage. So this protonemal stage is known as juvenile stage. From this protonema, prostate stage, this leafy gametophyte is developed. So after development of the leafy gametophyte, this leafy gametophyte develops or produces the sporophyte at its apical regions. So if we observe the mature plant body is divisible into main axis. It is looking like stem of higher plants. On this stem axis, leaves are there. Leaves are generally simple. They are ovate. They are spirally arranged on this main axis. The basal portion of the stem, which is underground, it produces the rhizoids, which take part in absorption of nutrients and water, as well as fixing the plant body firmly on the substrate. The main shoot axis may be branched. It produces two types of branches. One type of branch, main axis branch, it can develop the antheridial head. Lateral branch can develop archegonial head. That means male and female reproductive structures are developed on these two types of branches, lateral branches on this leafy gametophyte. Afterwards, sporophyte is developed. This sporophyte in Funaria is divisible into three distinct regions: foot region, seta region, and capsule region. So I have drawn here a single leaf of Funaria. Leaves are sessile ovate in shape. They are having acute apex, entire margin, and absence of lateral veins. Only single midrib is there. Lateral veins are totally absent. Leaves are small, scaly, sessile, having single midrib, acute apex, entire margin, etc. If we observe the rhizoids, rhizoids in Funaria are extensively branched and they are multicellular. We have already studied the Rixia. In Rixia, two types of rhizoids are there, tuberculated rhizoids and smooth-walled rhizoids. Both rhizoids are unicellular. But here, rhizoids are multicellular, extensively branched, and they shows oblique septa. That's all about the morphological characters, external morphological characters of this Funaria. Then, if we observe the transverse section of main axis of Funaria, it shows three distinct regions. First region is known as epidermis, second region is known as cortex, and third region, central region, is known as central stand. So first region, epidermis, is the outermost protective covering. It is made up of thin-walled, compact layer tubular cells containing chloroplast. Epidermis is without stomata, as well as it is lacking the cuticle. Due to absence of cuticle, due to absence of stomata, the epidermis layer can absorb the atmospheric moisture directly. Epidermis layer possesses chloroplast, and due to presence of chloroplast, the epidermis layer of main axis or stem axis also take part in photosynthesis. Epidermis is followed by cortex. Cortex is made up of few layers of thin-walled, compact layer parenchyma cells. If stem is young, main axis is young, the cortex cells also possess the chlorophyll pigments. When stem axis becomes mature, the cortical cells becomes thick-walled. When it is young, they are thin-walled and containing chloroplast. Central zone in the main axis is made up of elongated, thin-walled cells, and this zone, central zone, is commonly taking part in the conduction of the water as well as nutrients. That's why it is similar to the conducting tissue stand of higher plants, but it is lacking the differentiation of xylem and phloem tissue like higher plants. Only parenchyma elongated cells are there, which are thin-walled one, but they are having ability to take part in transport of water and nutrients. If we take the transverse section of leaf of Funaria and observe under microscope, the anatomy of leaf is very simple. Leaf doesn't shows tissue differentiation as like this main axis. There is a single layer of cells. These cells are having more chloroplast. The layer of the cells are also lacking stomata as well as cuticle. At the middle region, 2, 3, 4 layers will be there, but at the lateral lobes of the leaf, only single layer is there, single layer of cell is there, and these cell layers are without cuticle as well as this epidermis layer is without stomata. Central place of leaf is occupied by midrib. Midrib contain somewhat thick-walled cells which take part in conduction process. So due to absence of stomata, leaf can take part in absorption of atmospheric moisture as well as atmospheric gases like CO2, etc. So that's all about the anatomical details of main axis of Funaria and TS of leaf of Funaria. Then the methods of reproduction in Funaria. Funaria reproduces by both types. It can reproduces by vegetative methods of reproduction as well as it can reproduces by sexual method of reproduction. So if we observe the vegetative methods of reproduction in Funaria, Funaria vegetatively reproduces by these 4 different methods. For example, number 1, fragmentation of primary protonema. Number 2, fragmentation of secondary protonema. Number 3, formation of gymma bodies. And number 4, by the apospory. So, first method, fragmentation of primary protonema. So, protonema is a prostate structure. It is extensively branched prostate, green colored structure. It is developed after germination of spores. That means in this capsule region of the sporophyte, spores are developed. When spores are liberated, after germination of spores, the prostate extensively branched structure is developed. It is immature gametophyte. It is juvenile stage in the life cycle of this plant. This prostate body, plant body, is commonly known as protonema. That means protonemal stage and lipid stage. These are the 2 stages. First stage is produced in the life cycle of Funaria by the germination of spores which are produced in this capsule of sporophyte. So when spore germinates, protonema is developed. So this protonema which is produced by the germination of spores is commonly known as primary protonema. So the primary protonema sometimes breaks and produces the many fragments due to death of cells at different intervals, at regular intervals in the primary protonemal filaments. Due to death of cells at intervals, the fragments of the protonemal filament takes place, and these broken filaments or fragments of protonema contains chlorophyll containing living cells, and these chlorophyll containing living cells are further responsible for development of again protonema, and from the protonema buds are developed, and from these buds the lipigametophyte of Funaria is developed. Then second method of vegetative reproduction which takes place with the help of fragmentation of secondary protonema. So secondary protonema is not developed from germination of spores. That means sometimes protonema may be produced without germination of spores. So this protonema is commonly called as secondary protonema. So lateral buds are developed on this protonema, secondary protonema, and from these lateral buds the lipigametophyte is developed. Then next method is formation of gymma. The gymma formation takes place from the terminal cells of protonemal branches. So terminal cells in the protonemal branches, they may divide in successive planes, in various planes, and form simple bill or gymma bodies, and these gymma bodies under favorable condition are capable to produce the lipigametophyte of Funaria. Last method of vegetative reproduction takes place with the help of apospory. So apospory, meaning of this term apospory, it is phenomenon of development of gametophyte from sporophyte without formation of spores. That means gametophyte is generally developed by the germination of spores which are produced in this capsule. Capsule dehisces, spores are liberated. When spore gets suitable condition for germination, they germinate and produces the gametophyte. So sometimes the development of this gametophyte may take place from sporophyte without formation of spores. So how it is possible? This is possible because when sporophytic tissue, especially stolon tissues in sporophyte capsule or seta. So this middle portion is known as seta, this portion is known as capsule. So sterile cells or sterile tissues in capsule as well as seta. Due to development of wounds in sterile region cells of the capsule as well as seta, protonema may be developed, and the buds are developed in this protonema filaments, and from these buds the leafy gametophyte may be developed. So that means apospory is the last method of asexual reproduction in Funaria. So that's all about the vegetative method of reproduction in Funaria.